Surabhi, time to start.
Hi, so am I audible to everyone?
Yes.
Hello. Hello everyone, and welcome you all to this session today, and where we're commemorating the International Day for Women and okay, I'll just center myself in the camera. Okay, so again, welcome you all. So today we're here to commemorate the International Day for Women and Girls in Science. So as you all would have felt at some point in your life that the women in the field of STEM are underrepresented, and there does exist a gender gap in the field of STEM. And so this is an initiative by TIFR to talk and even out that difference, and we are and to encourage women and girls to pursue science if they have an inclination towards it. And towards that, today we'll have some really wonderful women talking about their journey in science and how they like if there were any challenges in their life in pursuing this career path, how did they face it, and what do they have to tell us about this field of what do they have to tell us about their research work. And so we'll hear a lot from them, and I'll introduce the speakers to you. Before that, I'd like to introduce myself. I am Surabhi Sinha, and I'm a PhD student in Tata Institute of Fundamental Research, where I work in the Department of Nuclear and Atomic Physics, and my work centers around studying electron-molecule collisions and like understanding the dynamics of those processes. And so I started my PhD here in 2018, and before that, I did not really belong to the field of research. I was originally an engineer in electronics and communication. And then I decided to make a transition into this, the research field. And it was not the most easy of transitions and decisions, but it was the ease of applications to the research institutes in India, and made it a bit simpler for me. So this is about myself. Now I'll give over the stage to Sneha, who will be the first speaker. And Sneha is a student. She's a PhD scholar in the Department of Biological Sciences in TIFR, and she works on studying mitochondria. And so she works on studying the importance of mitochondria in neurons, and we'll hear a lot more about this from her herself. So let me give over the stage to Sneha now. And hi, Sneha.
Hi, Surabhi. Thank you so much for that kind introduction. I'll just share my screen. Is my screen visible to everyone?
Yes, Neha.
Okay, great. Good morning, everyone, and happy International Women and Girls in Science Day to all. I'm Sneha Hegde, and I'm a PhD student in TIFR Mumbai. And today I'll talk a bit about how I came to research, what was the journey that led me to do a PhD over here in TIFR, and a bit about my work and my experience as a PhD student, or a junior scientist, as I'd like to call myself. So my journey towards science actually began when I was in school over here. I am a Mumbai girl, as they like to call me over here. I was born and brought up in Mumbai, and I studied in St. Paul's in Dadar, and it was during this time when I was in one of my science classes. When I was reading one of the chapters, I realized that I really like doing science and looking at how the scientists have done experiments to discover something that is in nature. It sort of gave me a very cool detective feeling, which encouraged me to pursue my path as a scientist rather than the conventional medicine or engineering career in the field of science. So this was further encouraged by a lot of the by all the teachers that were there during my time. As you can see, so I was so this my school was a convent school. It was an all girls school, and all the teachers were also all women teachers, which inspired me to show that being a woman or a girl is not going to be an obstacle to achieve whatever I want. They were quite driven, and that was like an initial inspiration from me as a childhood, as a girl, to do whatever I want to and not just stop myself and oh my god, I can't do this and I can't do that and so on. So yeah, after my school, so since I set my mind that I want to do science, obviously the next step was to go ahead and take science as a stream. So I did my 11th and 12th in the SIS College of Arts, Science and Commerce, where I pursued science as where I took physics, chemistry, biology, and psychology instead of maths, because I was interested to see what this other psychology subject is. And further on, I was sort of curious to see what this new, so biotechnology was sort of a new upcoming field at that time, at least in our area. So I was quite interested to see what this biotechnology field has to offer for me. So I decided to stay in the same college because it provided a good biotechnology program course, and pursued my bachelor's of biotechnology in the same college. Now, during these years of my junior college and my biotechnology and my bachelor's field, I was privileged to meet a few wonderful professors who I call my mentors now. One of them was Dr. Fani Kumar, who I met during my junior college years in my classes. And when I shared my aspirations of doing my PhD in science and being a scientist rather than being a medicine or an engineering person, he was quite excited and sort of encouraged me and pushed me to the directions or towards thinking of how a scientist would be, what a scientist would think about. And he sort of started training me from that point onwards as to think about problems, think about searching problems, even in places where where you think that there won't be any problems existing over there, and trying to find out solutions to those problems. Apart from that, during my bachelor's, I met these two wonderful women, Dr. Raj Lakshmi Amudan and Dr. Tara Menon. Both of them are biochemists, and one is an immunologist. And they sort of gave me the opportunities to explore about the different programs that are present all over India in the field of biotechnology, because I was doing my bachelor's at that time in biotechnology, such that I would progress in the scientific field in the same background, but do a PhD in biotechnology. And they all of these three mentors sort of helped me to shape molded me to think in a scientific way and not only in the applicative way. So that was a really good start and push for me for my scientific career. Then moving into the paths, under their guidance, I then applied for the central exam for biotechnology because I liked studying the course in my bachelor. So I wanted to pursue the same course, but in a very specialized manner. Hence, I wrote an exam, which is the central exam of biotechnology. It's a pan-India exam. After which I chose to be in MSU, which is in Baroda. It's Maharaja Sayajirao University of Baroda. And I chose this university primarily because the biotechnology program that they offer is amazing. It's research oriented, and the professors over there sort of trained me in a very rigorous manner to think scientifically, to navigate my way through places where people think that, okay, this is a roadblock and you can't go ahead. Over there, again, I had the privilege to meet these wonderful professors whom I call my mentors now, Professor Mrinalini Nair and Professor Johannes Manjrekar, one of which is a geneticist, Professor Mrinalini Nair, and Johannes Manjrekar sir was a developmental biologist. And both of them made sure that that there are no loose ends remaining in my way of thinking, such that once I leave MSU, I would be a much better scientist than I entered MSU over there. And obviously, I was encouraged and pampered a lot by my batch. This is the 2016-18 batch of biotechnology over there, wherein all of us had come from different parts of the country, and we were staying together and doing experiments together. And the lab, the background that you see here is our lab, and this is the lab where I first held my pipette and actually started pretending sort of to be a scientist, and we developed as scientists as a whole, all of us together. So MSU, I would say, helped me a lot to lay down my foundation as a scientist, and gave me sort of the confidence that, okay, this is the field that I want to be in. But I wanted to explore what fundamental research is, because my background so far was an applicative scientific field. So that's why I wanted to shift into fundamental research, trying to understand what is happening in biology at its basic levels, because of which I gave the exam for the GS exam for TIFR. And I got over here, beautiful campus, and it's like heaven for research, because you have all of the opportunities, research opportunities that are available all across the country over here with no inhibitions, and you can just pursue your research just in a very unlimited manner. So once I got into TIFR, I chose to be in Professor Sandhya Kaushika's lab. Sandhya is a cellular neurobiologist, and she tries to see what is happening inside the cell of this round worm, a worm called C. elegans, whose introduction I'll give later in my slide. By just not cutting off the animal or not harming the animal in any way. So this sort of excited me, because it would allow me to actually gain more exposure to this field of fundamental research, especially neurobiology. And hence, I decided to do my PhD by having Sandhya as my PhD supervisor. And I'm really glad I did that. So as a scientist or as a PhD student, what do I do? As I said earlier, it gave me a cool detective feeling. So we are actually detectives. What we do is we mark the objects. So biologists or cellular biologists, what normally do is we mark the objects or processes of our interest by different proteins or dyes, which I'll show in the upcoming slides. Then we change these processes in certain way to see whether the objects that we are looking at are involved in those processes. And then we just observe these changes. So where do I do these changes on? In my lab. So the first image that you see is a molecular biology lab, where we do all the modifications in the DNA or build the reagents that we want to. And the second image to your right that you see is something that we call as a C. elegans facility, where we handle our model organisms and do all the actual experiments. Now, I've been talking about C. elegans in the past couple of slides. So what they are the systems or the organisms where we do these changes. These are basically round worms, small worms that are present in the soil, and you can easily isolate them from your garden. Or if you just keep a rotten fruit and keep it rotting outside for a couple of days, you will find these worms present around these rotten food stuff or fruits as well. The advantage of having C. elegans as a system for research is because, as you can see here, it's a transparent worm. So anything that is inside the body of this body of these animal can be easily seen. And what we over here do is that we just express a particular protein that literally glows in the dark, and we see the cells by just shining some kind of light onto these animals, which I'll show in the upcoming slides. It helps us to see what's happening inside the cell live. It's like taking a movie of an entirely different world, which is happening within the organism itself. So that's quite cool. And because they are smaller in size, the longest that this worm can go on is about 1 millimeter. That's why we need a microscope to handle with them. And this is how you'll see animals within the microscope. So there are many of them. Since they are small in size, you can just there's no space constraint for using them, and it is quite easily it can be quite easily handled as well. How do we see the changes? As I said earlier, we use the advantage of their bodies being transparent. We express certain proteins within the cells of these animals, and then we just use a microscope, which is shown in the picture, to magnify these cells under the microscope, shine some amount of light, and voila, we just see some glowing worms with glowing cells within the body of these worms. So the picture that I'm showing below is actually an entire worm with certain neurons that are specific that have these glowing proteins being expressed within their body. And this is literally how we sort of track all the changes and different processes that are happening within the cell of these organisms. So C. elegans is not just restricted for to understand to see what is happening within the body of the animal. We can also do some cool behavioral changes. So there's one advantage of this is that, so for example, if we undergo certain changes, it is reflected in the form of behavior, right? So if we are thinking about something, we are just in a sort of different zone. If we are sad, we have that different emotion. If we are happy, we generate a very vibrant and positive energy all around us. So just like there are certain changes is reflected in the way we as humans behave, in the similar manner, any changes that we do in these organisms can also be, if they are important for the animal, that will also be reflected as in a way that in the behavioral changes of these animals. So one of the behaviors that I do is simply touching the animal at the head and the tail, which can be seen from the video here. And they just instantly respond to this slight touch at the head and the tail of the animal. So if the changes that I'm looking, if that I'm producing within the animal is quite important for the animal to move, it will just not respond to this touch at the head and the tail that's provided to them. So the cells that I work on, because I'm a cellular neurobiologist, the major cells that I work on are neurons, which are essentially the most important and the basic unit of our brain. These neurons are the ones that communicate the information from one place to another, which in turn helps us to talk, raise our hands, or something which can be as simple as just walking or putting a step forward from one place to another. I am more specifically interested into this small organelle called mitochondria, which I'm sure everyone knows to be the powerhouse of the cell. And if I zoom in these mitochondria from the neurons that I work on, you can see that there are a lot of them along the neuron or along the axon, which is like the major process in the neuron. And most of them are moving, and most of them are stationary, that is, they are just there lying around, and some of them are moving. So when I saw this image for the first time, I was quite interested to see I was quite curious to know that why are there so many organelles or so many mitochondria in the neuron, firstly, and what is the importance of having these organelles all throughout the body of the neuron? And that is basically what my PhD thesis is. So what we normally do, what almost all the scientists do, is that we use a system where things are completely fine, which can be seen through the image that's shown here. And then we use the system in which we either remove mitochondria and remove the object of interest and see whether there are any changes within the physiology of the animal, which can be seen through the image that is below here. So one small primer as I said, that we can do behavior on these worms. And what I see is that in animals which have mitochondria present throughout the body of the neuron, the animal is behaving properly. It responds to touch stimulus properly. But in animals which do not have mitochondria along these axonal projections, again, this axon is basically the projection that helps the neuron to communicate to its neighboring neuron. the animal just simply does not respond to the touch stimulus that's being provided to them. It just moves on like nothing's wrong. So this sort of suggested to us that mitochondria are quite important for the physiological, have a high physiological relevance for its functioning, for something which is as simple as locomotion. And currently what I'm trying to do is try to see how mitochondria are important for these neurons at the cellular level and at the behavioral level. What changes does it bring about within the system such that it helps the animal to respond, to at least sense that the touches, that it's being stimulated at the head and the tail region. Apart from that, our lab also does some cool stuff, which is again imaging, which is like taking movie of the neuron. So I would like to call these cells as a city, and just like we hustle and bustle within our city, there are a lot of things within objects within the cell that keep moving around doing their day-to-day stuff. And what we do is like just like movie directors or camera person, we just focus on one particular process and just take the movie of that particular process happening. So the movie that I'm showing you at the left is that of microtubules, which you can consider as railway tracks happening throughout the neuron. Railway tracks which are present throughout the neuronal process. And you can see they're quite coolly moving around, doing their usual stuff process that is transporting things from one place to another. And this is another movie that is taken by my another colleague of mine from my lab, which is a movie of a vesicle, which you can consider them as the trains which move along these tracks of microtubules. And as you can see, it's just never stopping. They just keep moving from one place to another. So that is why it's quite amusing for me to see them, because it's like a city, and there are people and objects moving around all throughout the cell doing their usual stuff and processes. Apart from that, as a part of my scientific career, I have had the opportunity to interact and work with one of the most brilliant scientists from all over the places, having healthy scientific discussions with them, trying to collaborate with them, and so on. I've also gotten the opportunity to attend scientific conferences both in India and abroad, where I had the opportunity to talk about my work, expose myself to all of these brilliant works that are happening all over the world, and gain a lot of experiences and, just interact with all of these scientists. And the most important part of my scientific career so far are all of the outreach activities that I have done, which sort of gave me the opportunity to convey my work out in the public, which also helped me to give a perspective about what I'm doing, why I'm doing, and how it is relevant. And there is a different joy to just, explain your science as a biologist to everyone else around you. And last but not the least, this is my lab. We work continuously as well as we enjoy after we do our work. This is after one of the laser tag parties that we went on after a long day of work. So yeah, that's pretty much it. I hope that I've done a good job. Thank you. And I'm open to take any questions.
So thank you so much, Sneha, for sharing your journey with us and talking about your work. It's so interesting. And so I think we'll take the questions later if there aren't any at the moment.
Sure.
Okay. So so thank you again. So now I'd like to invite the next speaker, Ruta ma'am. She's been a scientific staff in TIFR for about 20 years now. She works in the lab of crystal growth, where her day-to-day work involves growing metallic and oxide crystals using different crystal growth techniques and studying the properties of these crystals at temperatures as low as 2 Kelvin. Apart from being a wonderful scientist, she's also an avid lover of poetry. And let us hear more from her now, and not just about the amazing work she does, but also about her journey as a scientist and a mother. So let me invite Ruta ma'am now.
Hello, Surabhi. Thanks for the nice introduction. Hello, am I audible? Yeah, okay. Thank you for the nice introduction. Now I will share my screen. Hello. So first of all, let me wish you a very happy International Day of Women and Girls in Science. And let me thank TIFR Outreach Committee for giving this opportunity to talk to you. So Surabhi has already told that I'm working in crystal growth lab. So we are day-to-day work is to grow the crystal and study their properties. So first of all, I will like to tell you about what I do in the lab, and then I will tell you how I came to TIFR, my journey till now. Crystal growth, basically, it is an art, science, and technology. When you say crystals, what comes to your mind directly is diamonds and jewels, something which has definite geometric shapes, which are very glittery and beautiful. So this is what you will have in mind when you hear crystals. But crystals, importance of crystals is not just for the aesthetics. So if you see the world crystal production, only 5% of the crystal production is in jewelry industry. The rest 95% crystal production is for the technology. So it is very important to grow very good quality, nice crystals and explore different materials which can give you different new technologies. So as you are familiar, you are all using gadgets, electronic gadgets, laptops, mobiles. All these have very small silicon chips inside. These silicon chips are made from the single crystalline silicon wafer. So if you have very good silicon wafer, then you can fabricate good working devices on it so that you can have good working instruments and gadgets. Then there are applications in other industries like lasers, then scintillators. So if you see this small crystal, this is the calcite crystal. So it has a property that if you incident a laser beam on one of the planes, then it has different refractive index along different planes, so that beam splits. So like this, the crystal has different properties along different planes, and that is what we explore to make use of it is in the technology. Crystals are not just in the lab or in the industries or in jewelries. Crystals grow inside our body also. So you are all familiar with the kidney stones. So there are calcium oxalate, uric acid, calcium phosphate crystals in the urine. So our body also grows crystals. There are some good crystals also in our body. In our ear, there is called calcite crystal that helps us balance the helps us maintain the body balance. So our body also grows crystals. Crystals are very big. Some of the naturally found selenite crystals are found in the caves. Then these sapphire and silicon crystals, these are grown in the industries. Then these are the molybdenum disulfide crystals, which we have grown in our lab. They are so small. You can see the length scale is just 2 micrometers. So crystals can be very small, they can be very big. With the brief introduction of what are the crystals, now let us understand actually what is inside the crystal or how the crystal is microscopically seen. So when we build the wall, the building block is a brick. So we arrange the brick in particular manner and we get the wall. So similarly, any crystal is made up of the specific brick, which is known as a unit cell. So there are 7 different crystal systems and 14 Bravais lattices. which make the unit cell for the material. So each material has a different unit cell, and this unit cell, when you repeat in 3 dimensions, you get the crystal. So this is the pyrite ore, FeS2 unit cell, which is a cubic unit cell. If you repeat this unit cell in X direction, you will get the line like this. If you repeat it in Y direction, then you will get the sheet. And if you repeat it in Z direction, you will get the entire single crystal. So this is how the single crystal is inside. There are no gain boundaries. If you stand anywhere within the crystal, you will see the same background all over. So another example is a common salt. See, crystals are in our kitchen also. Sugar and salt are also the crystals. So NaCl has a FCC structure. If you repeat it, you get the NaC crystals. So why we are studying the crystal structure of the materials or studying the property? Because how the atoms and molecules are arranged inside the material decide the properties of the material. So if you see these coins, there are 35 coins arranged in the 7 rows. So they are left-hand side and right-hand side, they are arranged differently. So this is known as a polymorphism. The same atom arranged in a different fashion will give you different material and properties. So this, for example, this carbon arranged in the cubic form like this gives you a diamond, which is very shiny and transparent material, very hard material. The same diamond, when it is in graphite form, it is very loosely bound interlayers, and that is used in pencil. So that is very soft, opaque, and black in color, the same carbon. So we are using it in pencil lead because it is so soft that when we rub it on the paper, the layers separate out, and that is how we can write with the pencil. The another thing is single crystal and polycrystal. So single crystal, we have seen how it is built. The polycrystalline material, which we see all around, copper, aluminum vessels. So all these are polycrystalline. The same polycrystalline material we can grow in the form of single crystal. So when they are in polycrystalline form, there are extremely small crystallites, many crystallites within that, but they are arranged in random fashion. So there are grain boundaries, and because of these grain boundaries, we get different properties. But if we grow the single crystal, then we get different properties. So this is the alumina crucible, which you can see it is a polycrystalline and it is opaque. But the same alumina, if you grow in the single crystalline form, then that is known as a sapphire wafer, and this is very much transparent. And if you add very small ppm level impurity to this sapphire, you can get ruby and blue sapphire. So this ruby and blue sapphire you are familiar with. But sapphire originally, the very good quality defect-free crystal is a transparent crystal. So 1 ppm or less than that level of impurity of chromium or iron makes it ruby or sapphire. So these things are used to tune the material for the specific applications. So if you add some impurity to the crystal, it can it can become good electrical conductor or thermal conductor. So this is how we are employing the defects in the crystal to make it better, or removing the defects to make it better. So how we grow the crystal in the lab is may sound you slightly difficult process. So first, let me explain the very simplest process where you can grow crystals at your home also. So I will explain how to grow the copper sulfate crystals. You can take sugar or salt instead of copper sulfate. So you take a container, clean container, take water in it, add copper sulfate and dissolve. So copper sulfate dissolves. You continuously stir it and add more and more copper sulfate. At one point, you will see that copper sulfate is not dissolving in the water. So that is the saturated solution. Now this saturated solution, you heat it to 50 degrees Celsius. At 50 degrees Celsius, the undissolved copper sulfate gets dissolved in the water because the solubility of the solvent solute depends on the temperature. Now you add more copper sulfate and keep adding and stirring it till you cannot add any more copper sulfate. So at this point, you have a supersaturated solution. This supersaturated solution, then you filter out in another clean container and then allow it to cool slowly. as you allow it to cool slowly, the temperature of the solution decreases, the solubility decreases, and extra copper sulfate, which is dissolved inside, it will precipitate out in the form of crystals. So you will see many small crystallites at the bottom of the container. So if you want to make a bigger crystal, then what you have to do is you pick one of these small crystals, then again make the supersaturated solution. You hang this crystal inside the supersaturated solution, allow it to cool. Now when it cools, copper sulfate comes out of it. And since you have added the seed in that, the copper sulfate which is coming out gets itself attached to it preferentially attaches to the seed, and the seed grows into a bigger crystal. So that is how you can grow the bigger crystal. Now what we actually are doing in the lab. So we are making the crystals of intermetallic compounds to study their properties, to explore if the material is useful in some new applications. So for copper sulfate, we are taking water as a solvent. Metals will not dissolve in the water. So what we are doing, we are taking the low melting point metals as the solvent. So we can try aluminum, gallium, zinc, indium, tin, bismuth. These low melting metals we can think of as a solvent for our intermetallic compounds. So for growing these crystals, copper sulfate you can take any container, but for growing metallic crystals you have to go to higher temperature. So you have to select the container appropriately. So that container should withstand that 1000 degrees Celsius temperature. Then it should not decompose before that. It should not react with your metallic compounds or solvent. So all these things you have to take into consideration while you are doing the similar solution growth in case of intermetallic compounds. So let me explain you the one cerium AgSp2 crystal which we have grown. So when you want to grow the metallic compound in the form of crystal, you take the ingredients in proper stoichiometric ratio. We are using the alumina crucible. Alumina can withstand up to 1700, 1800 degrees Celsius temperature. So we are taking alumina, and alumina is oxide. It will not react with metals easily. So that is a good choice for the container. You take the metals, then we are putting the quartz wool as a filter on the top of it. And now I cannot heat it directly to 1000 degrees because metals will oxidize. So I have to seal it in the vacuum. For sealing it in the vacuum, I take the quartz tube. Quartz can withstand up to 1200 degrees Celsius temperature. So I insert this crucible in the quartz tube, vacuum pump it, and then seal it in the vacuum of 10⁻⁵ or -6 torr. This ampule then I keep in the box type furnace. I heat the furnace slowly to 1050 degrees Celsius, keep there for homogenization, and then cool slowly. So here we are taking for copper sulfate, we are taking water and just adding copper sulfate to see whether it dissolves or not. But these things we are doing it in the box furnace, so we cannot see if the solute and solvent they are in proper ratio. It makes a supersaturated solution or not. So this all comes from the experience. So generally the charge, that is CAgSb2, we call it as a charge, which we want to grow in the form of crystal. And we have used here antimony as the flux or the solvent. So charge to flux ratio we take as 1:19 or 1:10 or 1:5. So these things come through trial and errors. Some things are available in the literature, so we can make use of it. And some things we have to explore ourselves. So like this, we have taken this in the ratio, and at melting point of antimony is 630 degrees Celsius. So at 700 degrees Celsius, we have to take it out from the furnace and put it in the centrifuge to remove the liquid flux. This centrifuge is the special centrifuge. Within seconds, it reaches to 5000 RPM speed. So when we take it out from the furnace and put it in the centrifuge, the temperature should not fall much. Otherwise, the flux will solidify and we cannot separate flux from the crystals. So when we put it in the centrifuge, because of centrifugal force, this liquid solvent will collect in the bulb, and the solid crystals, they are filtered by the quartz wool and they remain in the crucible. Then when it comes to room temperature, we break the crystal, break the quartz tube and separate the crystals and study them further. So there is another method to grow the crystal, that is from the melt. So for the congruently melting compounds, you can melt the compound and you cool it slowly so that it crystallizes in the single crystal form. I will explain here. We have the very good 4-mirror optical float zone furnace where we can grow oxide crystals. Oxides generally have very high melting point. So we have to have a furnace which can go up to 2000 degrees Celsius. So during your childhood, you might have used the lens and the sun rays to burn the paper. So you are focusing the sun rays with the lens to the point, and locally there is a heating and paper starts burning. The similar principle we are using here, we are using 4 ellipsoid mirrors and the halogen lamp. The halogen lamp light is focused with the help of ellipsoid mirror to a 1 centimeter area. And that 1 centimeter area is a hot zone where you can go, you can change the lamp power and go up to 1800, 1900 degrees Celsius temperature. So in this case, we have to make the polycrystalline rods of the material which we want to grow in the form of crystal. So we have grown gallium oxide single crystals. Gallium oxide is a very new material and it has wide applications now in power electronics. It has a wide band gap. At the same time, it has good electrical and thermal conductivity. So it is a very interesting material. So we are growing this in our laboratory. So for making the polycrystalline rod, we have to put the powder inside the balloon, the normal party balloons, and then pack it and then press it in the hydrostatic press so that the powder compresses and makes a rod form. We separate it from the balloon and then sinter it in the furnace at 1400 degrees Celsius. Now we have a sintered very hard rod of gallium oxide. This gallium oxide rod, then we hang it in the furnace. We have a quartz tube where we can change the atmosphere, vacuum, air, argon, and that also changes the property of the crystal. So you hang the feed rod and seed rod, and with the help of lamp power, you can adjust the lamp power so that the feed rod melts. So I will show you here a small video how. Yeah. So the feed rod melts and it attaches to the seed rod, and we are continuously rotating the feed rod, seed rod, and translating it slowly with 1 mm per hour, 2 mm per hour, so that the melt which is on the top of the seed rod, it will slowly cool down and solidify in the form of crystal. More melt will come from the feed rod, which is translating down, and this is how we can grow a crystal. And this is the grown gallium oxide crystal, which is very transparent and very good quality. So this is what actually is my day-to-day life in TIFR. So how I came to this, right from the school, I will tell. My father was working in a bank. So every 2-3 years, we have to go to different places. So different schools. So my first school was the municipal school, because we were living in very small village where there was no water taps also. So we have to bring water from wells. So that type of village, there was only one municipal school. So my schooling started from there. And then later on, when I was in 3rd standard, we went to some taluka place, district's place, and then Mumbai, Pune, like that, my journey of the schooling came. But this interest in the science, it developed in those village schools, because there those teachers were so enthusiastic. They made us participate in the science exhibitions. They used to tell how to build the science models. They used to explain us how it works, and they made us present it in the exhibition. And that is how we grew interest in the science. So that doing the experiment and doing something in a research field, that thing came from there, the schools. Then I did my BSc and MSc in electronic science from Pune University. In Pune University, I worked under Professor Shashikala Gangal. She was my mentor for MSc project. And later on, I worked under her as JRF student also. So that time we were doing work on the silicon cantilevers we manufactured. And for that, again, the long experiments were needed. We have to first oxidize the silicon wafer. So we have to heat it, put it in the tubular furnace, pass argon gas, take it to 1000 degrees. Then for 2 hours, we have to pass oxygen gas again, pass argon gas and cool the furnace down. So how to work in the lab, how to plan the experiments, how to be a good researcher, that shaping Professor Shashikala Gangal gave me. She insisted that I should not take any advantage that, okay, I'm girl, I cannot wait long, I have to go home early. No, no. This is my experiment. This I want to do. So I have to stay. I was living in a big city like Pune. So I can stay there for long. There was no problem. So I should not take undue advantages. Then we have to use gas cylinders. So we have to transfer it within the lab or something. So every time I should not ask help from others that, okay, you connect the cylinder for me. I should learn it myself. Yeah, transporting cylinders is a risky job. So nobody should do it alone. But you can do it in presence of others. You should learn the techniques. The cylinders are heavy, but you don't have to lift it and transfer it. You can just rotate it and translate it. So these techniques, if you learn, you can do some kind of physical work, even though you have less physical strength. And some things, yeah, I agree that women have less physical strength. So that you accept it and you take help from others. At the same time, if they need help in any other form, who are helping you, you can help them. When I joined TIFR, I joined and Professor Dhar, and then Professor Tamivel joined our lab, and the crystal growth facility started. So the low temperature experiment Professor Dhar taught me, and the single crystal growth Professor Tamivel taught me. And I got very good mentors in TIFR also. When I joined TIFR, my baby was just 8 months old. So it was very difficult for me. I was living in Chembur and I had to come to Kulaba every day. I cannot, this traveling time is 1.5 hours one way. So 3 hours daily plus 8.5 hours duty. So I have to be away from my child for 11 hours. So that was difficult. So I used to carry him in local train to the office. We had a very good crèche there. So I used to keep him there so that even if he, in case of emergency, I can reach out to him urgently. So that was the very hard period. During that period, my mentors helped me, encouraged me, keep me motivated, doing the work in the institute and also looking after my child. So balancing these two things. So I'm thankful to you, Professor Dhar and Professor Samuel. Thank you.
So thank you so much, Ruta ma'am, for sharing such a wonderful talk with us and your journey in science. And do we have any questions?
We'll take them in the end.
Okay, we'll take the questions in the end. So then let me just introduce our next speaker to us, Radhika ma'am. She's a mathematician and a faculty in the Department of Mathematical Sciences in TIFR. She did her undergrad in engineering physics at IIT Bombay. She received her PhD degree from the University of Utah in USA, followed by postdoctoral positions at Temple University, University of Bristol, and Technion IIT. Her research is in the field of geometric group theory. She's, of course, an accomplished mathematician, and interestingly, she's also has an ardent love for sketching. So let us now hear more from her on her journey in science.
Hi, everyone. I'll just share my screen. I don't know why it's stuck. I'll try again. Okay. Happy International Day of Women and Girls in Science to everyone. And thank you to TIFR Outreach for inviting me to speak here. So I guess I should first tell you who I am. And in general, that's a very hard question. I'm still trying to figure out who I am. But for the purpose of this talk, let's assume, or I will say I'm a mathematician. And my journey started, so I'm from Jaipur. I did my schooling there in the same school from KG till 12th. And I was this kid who was always good at math, who liked doing math, science. I took science in 11th and 12th. And so my brother gave JE and came to IIT. So without giving too much thought, I was like, I like science. Let me also write JE. So when I wrote JE, that was the first time I felt that I was like in a class of 30, 40 students. I was one of like 4 women students so we would sit in a bench in the front of the class and then the whole class would be behind us. But we were so focused on studying at that point that it didn't matter too much. Then when I entered IIT, so that's when I realized I'm not that smart. Because in school, you like there is a bubble where you get 95% and then you think you know everything. But then when I came to IIT, everyone got 95% in school. So that was a good realization, that like learning early that there are way more smart people out there, but that shouldn't stop me from doing what I want to do. I think that was very important. So in IIT, I took physics, because when I was in 11th, 12th, I really enjoyed physics. Math also I always liked, but physics was a lot more fun. I think I really enjoyed mechanics problems and electromagnetic theory and all those things. But in my second year in IIT, I took a minor in math, and slowly I realized I was spending more time in math. Because when I was doing physics, I think my brain likes abstract stuff better than practical stuff. So when I was doing like in physics classes, there were equations to solve, and I could solve the equations, but I could never understand how they explain this physical phenomena. So I just naturally started getting attracted towards more abstract math. So I was able to do my final year project in math, which I think was very helpful for me to apply for grad school applications. So I went to University of Utah for grad school, which is in the US. That was also an interesting point. I had friends, like people are smart at IIT, right? So I had friends who were going to Stanford, Harvard, MIT, and I was going to this place, Utah, which nobody had heard. So for some time I doubted myself. I was like, why am I going to this place, which is in the middle of nowhere in a desert and nobody's heard of it? But to be honest, that's the best thing that could have happened to me. That place is amazing. I found the best advisor in my area of research I could possibly imagine. And that I think just changed a lot for me. So even though I thought I was not at a best place, but making the best out of the situation. And of course, there was luck involved. That worked out very well. So I said I always liked math and was good at it. But this was only when I started grad school that I realized I actually don't know how to do math. Because until then, I was just like solving a lot of problems, like algebra problems, or in undergrad, I was reading these books which had proofs and theorems. So I would read a statement, I would read a proof and move on. But in grad school, I realized like you have to, like there are no labs or there is no practical things to do in math, but in a way there are. You can work with examples. So like with my advisor, he helped me learn how to do math properly, actually. So in grad school is when I learned that. And then I finished grad school. I went, so I did 3 postdocs. So I think in sciences in general, it's becoming common to do multiple postdocs. So I first went to Israel for my first postdoc. That was also interesting. All my friends were staying in the US. I thought I'll also get a postdoc in the US, but I didn't. And so I went to Israel. My advisor told me that there's a great place there. And again, that was a blessing in disguise. That place was amazing to do research as a postdoc. They had so many other postdocs in my research area. Also, being in a place which was not India and not the US. Being in India, we know a lot about the US through all the TV and movies we watch. But being in a third country was a big eye-opener, just generally in life. So then I moved to UK for 1 year for a postdoc. Then I went back to US. doing these multiple postdocs, I guess one idea was to get exposure to different kinds of math. I worked on a particular problem in my PhD, but that's not where I wanted to stay for the rest of my math career. Like you want to broaden your horizon, like you want to work on different kinds of problems. So through these postdocs, I was trying to give myself exposure to different kinds of problems in my research area. I should say that my research area is, it's lucky in the sense that there are plenty of female mathematicians worldwide in this area who are at faculty positions. And so seeing them throughout my grad school and postdoc, interacting with them was very motivating for me. There are other fields of math where there's hardly any women. And I'm just thankful that I had people to look up to in my research area. And now here I am at TIFR. I joined last summer. So I just moved back to India last summer. And I'm enjoying my time here. So I'll share a few quotes that I like. So this one is by von Neumann. If people do not believe math is simple, it is only because they do not realize how complicated life is. So this resonated with me a tiny bit during the pandemic when things around me were so complicated. Sometimes it was actually easier to do math because there was some structure, there was some logic to what I was thinking or trying to do, but everything else seemed so chaotic. Another thing I like to keep reminding myself is, it is good that I don't know much. That way there's always more to learn. I don't remember who said this, but I've had this quote in my diary since I was writing JE. Okay. So I'm sharing this little picture from Marissa Loving, who's at UW-Madison. She made this really beautiful picture of what mathematicians do. Because I think that's always a mystery. People who work in labs are like physicists or biologists, chemists. Not all of them, but a lot of them work in labs, and people seem to think they understand what they do. But so what does a mathematician do? A lot of it is, so there's this bubble for math. Well, you have to do math. Oh yeah, I missed the perpetual coffee breaks, yes. So there's a lot of thinking. You have to do math. You have to think about proving problems, but you also have to find problems to work on. So a lot of like doing math involves reading research papers, like what is already out there. There are research meetings. So there's a lot of, at least right now, there's a lot of collaborative maths that happens. There are fewer people who sit in their room, close their door, and just do math on their own all their life. There's a lot of collaboration that happens. Teaching is a big part of being a mathematician. Well, teaching helps one actually learn stuff. When you teach something, you actually ingrain a lot of it. Then mentoring is a big part of being, I guess mentoring is a big part of being a faculty in any discipline. You have students to advise. They could be your PhD students, they could be undergrads. Then you have to write them letters. You have to run seminars from them. My favorite part, or after doing math, is traveling. As you probably noticed, as a mathematician, I got to travel a lot. Like I lived in 4 different countries while like doing my PhD and postdoc. So that was amazing. So you travel for conferences, seminars, you travel to work with your collaborators, you travel to give talks, and so on. There are more things in this picture, but I think these are the ones I wanted to point out. Okay, so that was a little bit about me. And now I want to tell you about the things I think about when I'm doing math. So it's another quote. So symmetry is a big word, I guess. And symmetry is one idea by which humans through ages have tried to comprehend and create order, beauty, and perfection. Right? We're always looking for symmetry in things. Chaos. Some people also look for chaos, which is also good, but our inclination is to look for symmetry. So an important problem in mathematics is to understand symmetries of objects. If I hand you an object, what you would do is you move it around in your hand and like your brain tries to understand it by understanding its symmetries. It's like what kind of objects? You could be thinking about regular n-gons. You could be thinking about higher dimensional solids, crystals, which came up in Ruta's talk. Like maybe I should say, like she did this example of carbon, like diamond and graphite. The symmetries of those two structures are very different. And that helps us understand that they have different properties as well. I mean, people also want to study the symmetries of the universe. And the idea is that once I understand the symmetries of an object, I can say that I'm closer to understanding what that object is. So let's just look at symmetries of a square. So what is a symmetry of a square? It's a rigid transformation. So I'm not allowed to squish and break the square, or I'm just allowed to take it in my hand and move it around. So I've labeled the corners A, B, and C just so that I can talk about them, but they're not distinguished. So how do I check how do I see how many symmetries are there? Well, one thing is I can take one vertex, and how many options do I have to send it to? Well, there are 4 vertices. I can leave it here. I can map it to the next one or the next one or the next one. So there are 4 options for a vertex to be sent to. So let's say I send A here, but then I have 2 options for the edges connecting A. I can either send B here and D here, or I can send D here and B here. So that's the times 2. So in total, I have 8 symmetries. And maybe I encourage you to take your pencil and paper and try to draw these 8 symmetries of the square. So it'll take you some time, and you'll do it because it's only 8. 8 is a finite number. But then a bigger question is there a pattern? Do you have a nice way of organizing these symmetries? Because what if I give you a shape with 20 sides? And then, of course, drawing that will be a pain, but you still want to be able to understand structure behind all those symmetries of that 20 gone. Okay, so here is a picture. So I just drew all the symmetries of the square, and maybe I'll point out one thing. So maybe let's just follow these colors. So if I go to this one, I've rotated the whole picture by 90 degrees. Here, okay, I think I didn't do it in order. See, like, that's what I randomly drew these, and they're not in order. But we all know I can rotate a square by 90 degrees, then 180 degrees, then 270 degrees, and then 360 degrees, I'm back. So here are my 90 degrees, 180 degrees, 270, and 360, I should be back. Okay, so let's try to put some structure to these symmetries. So let me look at the set of all symmetries of the square. And this set of symmetries. So how do I say what are the elements in this set? I say, okay, like symmetry 1 sends me from my original square to this square. Symmetry 2 sends me here. Symmetry 3, symmetry 4, symmetry 5, and so on. And there's an operation on these symmetries. I can compose 2 symmetries. I can do one symmetry, and then I can do the second symmetry on the result of the first symmetry. So I can compose symmetries, and they have some nice properties. There is always an identity symmetry, which means I don't do anything. That is a symmetry of the square. For every symmetry, there is an inverse symmetry. What is the inverse symmetry? Just undo whatever you did. If I rotated 90 degrees clockwise, just rotate 90 degrees anti-clockwise. So for every symmetry, I can just like undo it. For every symmetry, I have a opposite of it in this set. Oops. In this set. And finally, there's something called associativity, but let's not worry about that. So I'm looking at the set of all symmetries. I've put some operation on it, and I'm just observing that it has some nice properties. And if I want to abstractify this, I would say a group is a set of elements with an operation. So for instance, the set of symmetries with the composition such that these other properties hold. What do I mean by above properties? Such that if I compose two elements in my set, I get something in the set. And if there is an element which does nothing, for every element, there is a negative of it or inverse of it, and so on. So an easy example is the set of integers. So what are integers? I'm looking at the set of these numbers from minus infinity to infinity, all the negative integers, 0, and positive integers. And the operation I want is addition. So I have to check a few things to say that this is a group. I have to check that if I add two numbers, I still get a number, right? There is always a 0. 0 is there, such that if I add 0 to anything, I get the same thing back. Is there always an inverse? Is there always a negative? Yes. If m is in there, minus m is also in there. And associativity. But then, okay, here is a question for you, which you can think about later. Let's say I look at the same set. G is the same set, but the operation is multiplication. Now I'm multiplying two numbers. So then I want you to think whether that would still be a group or not. Like if you multiply two integers, you still get an integer, and so on. You should think about that. Okay, so what's the big mantra here? That every group, like if I define this group, if I define something abstractly as this set of elements with operations, blah, 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 blah. In my research area, we try to view every group not as this abstract definition, but as the group of symmetries of some objects. Because group of symmetries of some object is what we understand, like that's more intuitive. So I started with symmetries, I made the definition more abstract, but then I'm saying, actually, I'm going to go back to symmetries to understand it. But this abstraction actually helps in maths. So I'll give you one more example of how I start with something geometric, some symmetries, and then I want to show you how if I like, again, abstractify it, or if I like talk about it in terms of group theory, then how that helps me actually say more. So sometimes the algebra can help me say more. Just the geometric intuition is not enough. But sometimes to understand the algebra, I need the geometry. Okay, so braids. I'm guessing a lot of people here know how to make some kind of braid. The only one I know how to make, I never learned French braid or anything else. The only one I know how to make is the one on 3 strands, which like in school, I used to make 1 or 2 of those. So how did I make this 3-strand braid? So I'm going to draw a pictorial for it. So let's see, what did I do? I take the 3rd strand. I take it over the 2nd strand. The 4th strand, I don't do anything in the 1st step. I did do this. Then in the next step, I take this last strand and bring it over the middle one. And so on. And I can keep continuing, right? Like this is how we do the braid. Okay. So now let's say you are feeling creative and you make a complicated braid with your friend's hair. So like you do something. But then how will you know whether that braid will stay or not? What if it just like comes undone immediately or after like 5 minutes or something? Like you want to have an algorithm or a way to tell that, okay, I've done this beautiful braid, which I was really enjoying, and it shouldn't come undone. How will you do that? So now here pictures are harder. Like it's hard to keep track of this. So I'm going to put this in the perspective of groups. I'm going to look at the set of all braids on n strands. So I was doing 3 strands there, and the operation is stacking. What do I mean by stacking? So I have the green braid, and then I stack the yellow braid below it. So I do one operation and then the next braid. And like if I want to draw it continuously, this is what. So this is another braid. So if I stack 2 braids, I get a 3rd braid. So already something, there is hope for this set to be a group. So I've drawn a few more braids. This is the identity braid. If I take a braid, some complicated thing, and if I stack this identity below it. It doesn't change anything. And this is just an example, sigma 1, that is just the first braid going under second braid. This one is 3/2. And here I've drawn the inverse of sigma 1. So sigma 1 is 2/1. And if I do 1/2, that's the inverse. Because if I stack these two, maybe let's see. If I stack these two, let me draw them continuously. If I stack these two, you can clearly see that I can just slide the second one over the bottom one. So this braid will come undone. So there is an inverse for this sigma 1 that I drew. So this is an indication that this set of n braids with the stacking operation will be a group. Of course, like you can check all the other properties. And it's a theorem that actually to generate any braid, like you might do, let's say you start with 3 strands, and you might do something super, super, super complicated. In fact, you will make this complicated. You have only 3 moves to do, or 2 moves, basically. That's what I'm saying. 2 moves and their inverses. You can only bring like this one on top of this and this one on top of this, or under. So I'm saying these are the only moves, and using these 4 moves, you can actually make any complicated braid you like. But then the question is, like, going back to the initial question, like if I make a complicated braid, I want to know that it will not be trivial. It will not come undone. So, and this is where group theory helps. So if I make a complicated braid, as I said, it can be written as a stacking of these sigma 1, sigma 2. So I write a complicated word, sigma 1, sigma 2. Let's say I keep doing sigma 2 100 times. Then I do sigma 1 inverse. Then I do sigma 2 10 times. And I do sigma 1 17 times. And I want to know, what happened? Like, did I get something concrete? So the question is this the trivial braid? Is this the identity braid? And using methods in group theory, you can actually come up with an algorithm. So this group is special in that there is actually a algorithm to tell you whether this word is trivial or not. So I've tried to give you 2 examples of like how, like the exchange between groups and geometry topology. And that's what I do in my research. This area is called geometric group theory. Sometimes I'm trying to understand groups and I use symmetries to understand them. Sometimes I already have a space and I want to understand it. And then I like attach some groups to it. Just some more examples of groups in real life. So crystals, I said, transformations of space or like universe, like people study that a lot. I didn't talk about this group. This is called modular group, which is used in cryptography. And even in phylogenetics, there are things in phylogenetics which use some group theory or some symmetry to understand. And they are like, groups are everywhere. Symmetries are everywhere. So groups are everywhere. And then finally, I leave you with this quote, which I also really like. And I always try to remember this. Whatever be the odds against you, fight, fight, and fight till the end. It makes a huge difference. Thank you.
Hello.
Hello.
Yeah, so there's a question for you, I think, on the group chat. So Anushka Tonapi wants to ask, so residue classes are also considered as groups?
Yes, so that's the one I did not really talk about, but let me show you. I have a slide on that, so like residue classes, like, so let's say I'm looking at 0, 1 are the residues from 2, right? The remainders when I divide by 2 are 0 and 1. And if I want to understand that set with the operation, so here I'm going to like attach some meaning to these. I have a switch. 0 means do nothing, and 1 means flip the switch. And the action is to compose these actions. So then do nothing just means do nothing. Do nothing and then flip the switch means flip the switch. Flip the switch and do nothing is same as flipping the switch. Flip the switch once, flip the switch again means you did not flip the switch actually. So this is like explaining the group action in this case. And then the clock arithmetic, the way we see time, that's like the modular group with respect to 12.
Okay, so I guess that has been answered. Are there any more questions? So we can ask. Yeah.
I have sent you on WhatsApp. Yeah, look at your WhatsApp.
Yeah. So there is a question by Abhishek Amar, right? Why our model organisms are very restricted? That is a question posed to Sneha.
So model organisms are really not restrictive. Different model organism has its advantages, and we, depending on what we want to use, we select that model organism. For example, C. elegans, the advantage is to just, we can check whatever's happening within them live in a single cell, at a single cell level. So that's why we use C. elegans as a model organism. So just like there's nothing as such called restriction. We make use of the advantages, and then the advantage, and then accordingly choose whatever model organism that we want to work with. I hope I answered that question.
Okay, so yeah, so I don't see any more questions on YouTube or in the chat. Yeah, so then if there aren't questions right away.
Actually, on YouTube, all are complimenting all the speakers of the day for the fantastic work, and wishing all of you very best. And I guess now it's all back to you. Back to you. Yeah.
Yeah, so why don't we, like, since the four of us are here, we can have a small discussion about some of the questions and all that we have as just scientists or women in the field of science. So I'd like to ask a question to Radhika ji. So you've talked about your journey as a PhD student and then as a postdoc fellow in different countries outside India. And now you're a faculty in India. So did you at any point feel an inhibition in returning back to the country? Do you think there are enough good research opportunities back in the country for math and science in general?
Thank you for the question, Surbhi. So I think for me, moving back to India was a mix of personal and professional both. So like I was saying, I went to Israel and then I saw that, like, okay, this is country which is like not India, not US. We are so obsessed with US. So I was like, okay, they're doing so well. It's a tiny country. And I was like India can do the same thing as well. Like we can be very strong in science and like research as well. So that was like the first time I felt more strongly that, look, okay, I want to go back and contribute to like the research side of things in India. And personally, it was my husband and my brother, like my family was generally moving back. I saw plenty of people around me moving back to India, which was very encouraging also. Research-wise, I think India has a lot of opportunities coming up. There are a lot of new IITs that have opened up. Some of them are really new, but like there's also a lot of ICERs which are doing really well. So those are really good places to come back to. I should say that being a theoretical scientist, it's a little easier for me to make that decision. Someone who's doing experimental sciences, I guess they have more things to think about. And I have some friends who have hesitations, but I also know some people who have made that move. So you really have to if you're an experimentalist, then you have to see what works for you and what. But theoretical scientists, I can just like move around with my paper, pen and laptop and work from anywhere. So that makes that decision easier for sure. I think, yeah, that's all I have to say.
I see. I mean, I think probably Sneha will have something to add to this, because I know that you work very closely with like opportunities for research students outside the field of academia as well. So yeah, like as Radhika ji said, that like experimental physicists, not just physicists, experimentalists may find it a little more challenging to find opportunities. So what do you think is the situation?
I think currently it's like India is doing a lot for research and development, even for experimentalists, right? From giving various kinds of grants to starting of new institutes or universities, just like Radhika ma'am just said, about for not only theoreticians but experimental scientists as well, giving the funding that is required to get the equipments that is needed, which was not there like a few years ago. So yeah, there are a lot of opportunities that are in pipeline right now for a lot of experimentalists as well to come back and settle in, because a lot of people have families in India, so they choose to come back to their motherland rather than staying over there. And it's becoming easier right now. That's what I saw.
That's good to know. I had some friends who were, I think, looking at this like 6 years ago, or like even before that. And one of them was not happy. But then I also know like other experimental physicists who have now moved back. So like, yeah, as Sneha was saying, probably things are getting better.
And are there opportunities in the industries, like for scientists? So just because, yeah.
Yeah, it's getting better. We are getting there. Actually, I've tried to talk to a lot of people in industry, as you just suggested.
Yeah.
Yeah. So as a student community, we are trying to develop this thing where we can bridge the gap between academics and industry people or non-academicians, so that the scientific community would grow right now. And we are getting there. There is a gap, but all of us, like all the scientific community from all the different parts of the country, are actively trying to fill in that gap by doing certain outreach events, or science communication events and programs and so on. So we are getting there. We are not there yet, is what I'd say, but there's a long way to go, but we've progressed.
Yeah. I mean, there's a community that is forming now that is looking to make it better. So that's nice. Yeah, even like Ruta ma'am, you've been in the field for so long, and so as having discussion with you earlier, where you said that there are some opportunities even from the government's end, to especially for women scientists.
DST actually funds some women scientist programs. So those women or girls who have break in their career after their post-graduation or after their PhD, they can use these schemes. They have to, suppose somebody has discontinued after their post-graduation, then they can register for their PhD, attach themselves to some lab, and then they can write the project proposal to DST. DST will conduct the interviews of such applicants, and they will select some candidates. Then they will be given fellowships, contingency, travel grants to pursue their career. Similar is the case with the PhD, post-PhD students. So this is very good initiative that DST is taking, because sometimes because of family responsibilities, after post-graduation or PhD, we have to take some break. And then again, coming back to career becomes difficult. So this is the stepping stone. Again, you can come back to the mainstream. And that previous question, like industries, yes, many of our department's PhD students, they got the jobs in the industries also. So that is growing. So industries here as well as abroad.
Oh.
So from science, you can go to industrial jobs also.
Nice. So that is actually very nice to know that you have opportunities outside just academia as well. And so it's nice to know it from so many different people, so now today we have as like a variety of people from different fields. So like, as you would have known it through the talks as well, but Ruta ma'am works in crystal growth and she is a scientific staff. Sneha is a PhD student in biological sciences, and Radhika ma'am is a faculty at TIFR for in maths, and myself, I'm a PhD student in the physics department. So it's quite diverse, and it's nice to be in a discussion with all of you today.
One more thing I want to add, that there are many IITs, ISERs, even TIFR have this internship program. So girls and women, they should participate in that. Once they actually work in the lab, they will know what is the atmosphere, what is the experiments are like, and they will not be scared of science or mathematics. They will be more familiar with that, and they will understand that science is not something abstract, that is in our everyday life, we are using it. So that is easy. It is not a difficult thing. So once they come to the lab, work in the lab, then that interest will grow. So they should take these chances. They should go for the internships and get themselves familiar more with the lab culture. That is my urge.
True. Yeah, if you have an inclination towards science, you should definitely consider it as a career opportunity as well, like a career option. So, and of course, as we were talking about it yesterday, there are things that may be physically straining for you to do, as you talked about, like moving cylinders, shifting cylinders from one place to the other in the lab. But there you may feel that you always require somebody's help to do that. But then there are other things that you can do which somebody else would not be able to do. Like, so there, like for example, as you work with electronics, you have to make some connections, electrical connections and all. So how delicately you make that. So there are things that do not have the like that do not require as much strength, and it's something that any of us can be really good at and maybe better. So
if the help is two-way, that means I am taking help from others and I am also helping others, then it is not a burden. And then you don't feel like, okay, I cannot do this, and I am always taking help from others. So you can find the ways, you can help the people who are helping you. And there are people, very good people here, they encourage you, they understand your problems. And it is very rare situation that they will taunt you or say you something. So it is encouraging. Actually, people are changing. They understand the things, and in the institutes like TIFR and many other organizations, the things are changing now.
True. So is there anything that we'd like to mention, let me think, or so I would at this note also take the opportunity to introduce the for the people that are attending over here, that some outreach program. So there is this Chai and Vai session on February 19 at Ruparal College in Mumbai, and the title of the talk is Ye Tara Wo Tara. So, and it's given by Aniket Sule and the team at HBCSE. And and another thing I'd like to announce is the National Science Day on February 26th, and where, I mean, people who are familiar with the National Science Day culture of TIFR, they know that they can just drop in and enjoy different experiments, visit around labs, and all other fun activities to do here at TIFR. So these are the two announcements that I'd like to make, and anything else if anyone wants to add. Otherwise, we can close on this note.
Okay, so I think Outreach wishes to thank all of you for giving a talk and conducting this session. And also thank you, audience, as well. And again, wish you a very happy International Day of Women and Girls in Science. Keep rocking. On this note, bye.